All right, good morning, everybody. Bag of stuff down over there. We have a pretty wild ride today because we have quite a bit left to cover before we finish. Right, so we pretty much finished up around here in terms of polypeptides, bonds between them, the characteristics of amino acids. Now we need to get on to actually making polypeptides, right? And so getting back to the whole components deal, obviously we've talked about the mRNA, right? Next point is we need to talk about the ribosome. And so the ribosome is essentially the thing that facilitates these tRNAs coming in the correct place, on a side, and catalyzes the peptide bond between the existing amino acid or polypeptide chain and the incoming amino acid that's being brought in by the tRNA. Okay, well, while we're here, might as well talk about this a little bit. So this is kind of a stylistic view of a tRNA. Two key areas that matter, at least in our context. There are other areas, but in our context, the ones that matter are the anticodon. So the anticodon is a series of 3 bases, just 3, right? And those recognize or base pair with the codon that tRNA matches to. Right, so we talked about how the tRNAs are the translator in translation. Well, this is how they do it, right? Going back to the ribosome, essentially you have 2 subunits. These are actually made up of a lot of proteins and several RNAs as well. We have the small subunit down here. This is responsible for binding the mRNA and moving along it. So the small subunit equals binding my time, mRNA, so initiation of translation, and processing along it. Right, so the small subunit doesn't have anything to do with peptide bond formation. So it's kind of like the motor or the carriage, right, that moves the whole caboose along. Now the large subunit up here is larger, as the name suggests, and has several particular features. So the large subunit, if I could spell that right, essentially, to simplify a great deal, is peptide, all about peptide bond formation. Oh, and also kind of, and reading the mRNA. And that's a little bit of a sloppy term. So the large subunit has a exit tunnel, right? That's where the newly synthesized polypeptide comes out. I'm just going to see if I can find, go away, bar. Nope, not that one. Here we go, that'll do. You get to see all my different tabs. All right, so it's gonna get back here. Kind of looks like so. Hopefully you've had time to check out these videos that are linked. There's one in the PowerPoint that I've added and uploaded. So there are 3 tRNAs present in the ribosome at this point. The amino acid chain or the polypeptide is being extruded out the top, like tube of toothpaste, right? And these 3 tRNAs are in essentially different states, right? One of them's coming in, one of them's staying, least for a little bit, and one of them's leaving. Right, and so when you look at the actual structure, it's obviously kind of stylistically done. You've got 3 different sites. The A site is the access, has a fancier name. It's the amino acid tRNA binding site, which basically means where the tRNA goes in. And it's a charged tRNA, right? That's what the amino acid tRNA means. It has a amino acid attached to it. And it's also where that tRNA base pairs with the matching codon on the mRNA. So that's where the tRNA goes in. Essentially, there's this constant flow of tRNAs going through the ribosome, getting recharged, and then coming back in again. Right. P site is pairing or peptide bond formation. And that's where the polypeptide is removed from the tRNA in the P site and added to the amino acid in the A site. And E is easy. It's the exit, where the uncharged, I don't know, discharged tRNA leaves the ribosome. So there's 3 different sites and different things happen in each of them. And those sites are key to how the ribosome works, how translation happens. Now, the part of the specificity of translation comes from the anticodon in the tRNA. In fact, arguably all of it does, to be honest. But also the specificity comes from the enzyme that puts the amino acid on the other end. So this is the amino acyl tRNA synthetase, which is a bit of a mouthful. And it recognizes both the amino acid very specifically, right? So there's one synthetase per amino acid, essentially. And it also recognizes the anticodon on the tRNA, right? So this is like the matchmaker. It is very specific for one amino acid and very specific for a tRNA. So it will only bring those 2 things together. So the correct tRNA gets the correct amino acid added to it. Because otherwise, the whole base pairing of the anticodon and the codon would mean very little, right? If you just stuck whatever amino acid onto the tRNA. So this is where the specificity, really comes into play. And so to do that, you need energy, right? So translation is very energy intensive, right? At several different points we'll get to in a little bit. The first of those is charging the tRNA, right? So that requires an ATP. So the ATP essentially kind of charges the synthetase with energy, changes its conformation, and the amino acid binds to this pocket right? And that pocket is very much specifically shaped to recognize that amino acid, right? Based on its charge, size, various other properties. Once the synthetase is charged with a amino acid, now the tRNA comes in and it fits like a whole hand and glove type deal. Right, you can actually see a space filling model of it over here. And again, this process is very specific. So when those two parts are brought together by the synthetase, the AMP, right, the kind of depleted energy form, leaves, and in the process, that bond gets formed between the amino acid and the end of the tRNA. As soon as that happens, it no longer fits quite right. The synthetase changes shape and the charged tRNA leaves. So even before translation has started, right, these tRNAs are constantly being charged, right, because you have to have a pool of them available whenever you want to translate an mRNA into a protein. So already we've used 1 ATP, right? So adding 1 amino acid to a polypeptide requires 3 units of energy. 1 ATP to charge the tRNA, 1 GTP, which is similar to ATP, to bring the tRNA into the ribosome, 1 GTP, to move the ribosome 1 codon along the mRNA. Oh, I did type that right. Right, so for just 1 amino acid to be added, you require 3 units of energy. Right, and so that's why, when you're on, an organism is under starvation conditions or nutrient limited in some way, or energy limited, translation is one of the first things that gets wound down. Because it takes a lot of energy. And given how important translation is in cell division and, cell growth and all that other stuff, now you can see why people from, famine-ridden countries or, places that have below average nutrition tend to end up smaller, right? Because they just don't have whatever their genetic background in terms of height and size and whatever, they don't have the resources essentially to do enough of this to allow them to grow to their full potential. So here's that video. It's real handy, right? Watch it if you haven't already. It does a good job of visualizing the process. Now what we're going to talk about is the initiation of translation, right? So the kind of similar in a way to transcription, the initiation is really where a lot of the kind of tricky stuff happens, right? And this is one example where bacteria and eukaryotes are quite different, right? They both have small and large subunits, right? As you can see here, they're actually different sizes. The ones in eukaryotes are a bit bigger, for whatever reason. More proteins, I guess. But there's some key differences. So one, prokaryotic mRNA does not have a cap. Instead, as soon as the start codon, this AUG, appears from the RNA polymerase, right? So transcription and translation are very closely coupled in bacteria. It's one of their kind of interesting quirks, I guess. But as soon as that appears, the small subunit can bind to it, right? And it does so using this sequence called a Shine-Dalgarno sequence. This is like a recognition sequence for the small subunit and this initiation factor. This initiation factor also stops the large subunit from glomming onto it, pardon me, until everything is in place. So actually for both prokaryotes and eukaryotes, the large subunit is what assembles last, right, in both processes. Okay, so again, as soon as that mRNA pops its head out of the RNA pol, the small subunit can bind to it, right? So it binds directly onto the start codon. And key point is this start codon, when assembled, and this is actually similar in some ways to eukaryotes, will be in the P site. So we will start with a start codon in the P site and a tRNA with methionine, which is encoded by the start codon, already present here. Right, so it's already kind of raring to go before anything has actually happened in terms of translation, at least the longer term process.
Professor,
okay, go on.
What did you say that the Schön-Gall sequence is that combines or helps combine the subunit to the larger unit or to the AUG?
Oh, so that's a recognition site. So the
okay.
Shine-Dalgarno sequence is a conserved sequence in the mRNA, just upstream of the start codon. And that's recognized by the small subunit. So it's basically a small stretch of mRNA sequence, AU, Cs, and Gs of some sort. I don't actually know what the sequence is, which says this is where you go to the small subunit. And so part of the small subunit recognizes that based on shape and sequence, I guess, and always assembles directly over the start codon. That help?
Yes, it did. Thank you.
Cool. Okay, so once that happens, then a whole bunch of stuff comes in, right? Including our tRNA or FMET tRNA. So that recognizes the start codon and is always the first amino acid of a protein. Now, not necessarily mature protein. I actually probably, I should have said polypeptide. There you go. That's probably a better way of putting it. So for every organism and every gene, every protein starts with a methionine. Bacteria, archaea, funky ass stuff, COVID, eukaryotes, all start with an AUG. So that tRNA is brought in with a bunch of other initiation factors. So IF equals initiation factor, and that's EIF in eukaryotes. God dang it. EF is elongation factor. Sorry, typing skills are not working very well today. And so their job is to bring in the tRNA. That takes energy. Here's that other, or the second unit of energy. And as soon as that tRNA is in place, now the large subunit can now assemble over the top of that FMET tRNA. So all these other bits and pieces leave. And now you have at the end, or the end of initiation, right? The beginning of translation, you have the ribosome assembled. You have a tRNA in the P site, and you have an empty A site. So now it's possible for a new tRNA with an amino acid to come into that A site. And I want to quickly show you, I'm going to keep my eyes on the time because I want to make sure we cover everything. I don't want to leave you short. You can marvel at my amazing number of tabs. All right, so this video is, where's that at? It's down here in slide 29. So just click on that, the purplish text, probably blue in your case, and that will open up that YouTube video. So essentially, it's just a series of steps. We've done the first one. We've assembled the ribosome over the start codon with a tRNA, an FMET tRNA in the P site. So really, the ribosome, particularly the large subunit, its job is to hold everything in the right orientation. And this region here is where the ribosomal RNA is. And the ribosomal RNA in the large subunit is what catalyzes the peptide bond formation. So the protein just kind of gives the scaffold, right? So it holds everything in the right place, allows things to come in and leave and so on and so forth. It's the ribosomal RNA that does the work, right? So this is after initiation has, kind of completed, right? Here's that Shine-Dalgarno sequence just upstream of the start codon. Now we have a empty A site, and in that A site we have a UCC codon, which I think codes for serine from memory. Let me just double check. UCC, there you go, that codes for serine. Go, back we go. All right, so now we're going to have a tRNA that's been charged with serine and has an anticodon which base pairs with that UCC codon. Okay, so what's the anticodon going to be sequence-wise? Great, tell me what it's going to be. What's the sequence of the anticodon that will recognize this codon?
AGG.
Yes, I should have worked that out beforehand, shouldn't I? Exactly, it's AGG, right? So let's kind of scooch along a little bit. Where are we at? There you go. So now we have our serine tRNA, and we have some elongation factors that will help that enter the ribosome. with the aid of some energy in the form of GTP. So that comes in like so. That GTP will be hydrolyzed to GDP and those will leave. And so now we have two amino acids very close to each other in the ribosome. We've got the one on the tRNA on the P site, and we've got the new one on the A site. So we need to do two things now. So once there are two amino acids close together in the A and P sites, we need to do two things. One, we need to remove this FMET amino acid from its tRNA, right? Because otherwise, imagine we'd just have proteins with a whole bunch of tRNAs dangling from them, right? So we have to cut that amino acid off. Right. And then second, we need to form a peptide bond between those two amino acids. I knew I didn't spell that right. Sorry, to have enough space, I can't actually see like the right-hand most part of the chat box. And that is all done by this peptidyl synthetase, I think it's called. Let's kind of scooch along a little bit. Yeah. Oh, peptidyl transferase. I'm sorry. Okay, so let's kind of go back a wee bit. Hadouk. All right, so it cuts that amino acid off, holds it in the correct conformation, right? And then forms the peptide bond with the serine, which you can now see here. So now we have a discharged tRNA. We have a tRNA in the A site, which has two amino acids attached to it. And now we need to move, right? And so now the whole ribosome moves one word along the mRNA, or pulls the mRNA one word through it, whichever perspective works for you. And so the next codon will be an AAG, right? What is that code for? AAG, lysine. There you go. So now comes the third unit of energy required. Let's kind of scooch. That just shows it in a nice kind of letter stick and ball, I think it's called. Go on. All right, here we go. So now we have our EFG, or elongation factor G, also with GTP, right? So the energy in GTP is what will move the ribosome one word along. It's like a ratchet almost. So that comes in here, right? See, there it gets hydrolyzed, the energy in that phosphate bond is released. Now we have moved one word along. So now we have, I'll move it just a little bit further. There you go. The original tRNA is now in the E site, no amino acid on it. The second tRNA is now in the P site, two amino acids. That's technically a dipeptide, right? And we have a free A site. You can guess what's going to happen now, right? It's not really that tricky. We're now going to have a lysine tRNA come in with our elongation factor and some GTP. And then we're going to do the same thing all over again. Come on. Oh, yeah, also the tRNA that's in the E site will leave. Right. Ba-dang, gone. And here's the lysine tRNA coming in. Just do the same thing all over again. Another elongation factor G comes in with some GTP, moves again. This discharged tRNA will be in the E site. This will be in the P site. And then there'll be another tRNA come in which recognizes the GCC. And that will happen over and over and over and over again until the ribosome hits a stop codon. Anybody got questions? So let me drink some coffee.
So the chains of amino acids that like accumulate, they just kind of eventually like get so long that they fall out or, like string out of the ribosome.
That's right. Yep. And then depending on protein and organism, that polypeptide will start folding into its correct conformation. So that's, where did that tab go? Here we go. So that's this kind of series of red blobs kind of coming down here. Also, as you can see here, it just gets pushed out one amino acid at a time. There you go. It's kind of weird listening to that at half speed. Some organisms, some proteins, this polypeptide chain is actually held kind of unfolded until it's all produced, and then it's allowed to assume the correct conformation. It just depends on the protein. So basically, in many ways, it's just the ribosome kind of allows these tRNAs to kind of flow through, essentially, and drop off their amino acids, right, according to the sequence of letters and words or codons in the mRNA. Okay, anybody got any other questions? All right, so those videos are well worth going through. They're really good, and they will help kind of visualize this process. Pardon me. So that's the deal with prokaryotes. So elongation is essentially the same in both prokaryotes and eukaryotes. The only differences are the names of the proteins that allow that process to happen, like elongation factors and so on. Right, so you don't really need to worry too much about that, but you really do need to understand the process. Now, one of the big differences between prokaryotes and eukaryotes is in initiation of translation. So in prokaryotes, the whole thing assembles over the start codon. In eukaryotes, the whole thing assembles way upstream in the 5' UTR, and it travels down, well, not the whole thing, the small subunit. This is a little bit of a misrepresentation. Travels down until it finds the start codon. Right. And so the proteins that help facilitate that are these 5' cap binding proteins, and also the poly A binding proteins as well, the ones we saw earlier that are involved in protecting the 3' end of the transcript. So it's a little bit more complicated than this, as one would expect. And so this is a much more accurate kind of description. And essentially, it's kind of, you could see some similarities, right? So essentially, a bunch of initiation factors bind to the 5' cap, right? They recognize its shape, its oddity, essentially, right? Because it's sort of RNA, but it kind of really isn't as well. Once those assemble, they then bring in the small subunit, right? So you can see it's actually quite a bit bigger. That small subunit complexes with a bunch of other initiation factors. And again, I'm not going to be quizzing you on what these numbers are, right? Because I can never remember. I just, if I need to know, I'll look it up. But I will be quizzing you on the process and the difference between prokaryotes and eukaryotes in this process. So we have a similar kind of deal. We have a FMET tRNA complexed with our small subunit and a bunch of other proteins. Difference is this is not over the start codon. The start codon is somewhere downstream. Could be several hundred base pairs downstream. So what we're going to do now is we're going to use this kind of like a barcode scanner. And this small subunit will process or scan along the mRNA until it finds a match. And when it does, ta-da, that's the start codon. So as you can imagine, there are no start codons in the 5' UTR. And even if there's an AU or a UG or an A something G, this tRNA is not going to recognize it. It has to be an AUG, nothing else. AUC, U, whatever, doesn't matter. Has to be an AUG. That's really basically shown here, right? So the complex forms, and now it scans along the mRNA until that tRNA base pairs with an AUG based on the anticodon of that tRNA. As soon as it does, a bunch of those initiation factors leave, and that allows the large subunit to assemble over that tRNA, right? So in many ways, this is, kind of analogous, right, to prokaryotes, because now the FMET tRNA is in the P site. The difference is how the small subunit and that tRNA gets to the start codon. In prokaryotes, it assembles directly. In eukaryotes, it starts at the end of the mRNA and moves down until it finds it. And essentially the same deal happens again, right? Now we have an open A site. Our next tRNA is going to be phenylalanine tRNA. Will come in, the methionine will get cut off of here, gets added to phenylalanine, ribosome moves along one word. Okay. Any questions? Okay. So really, elongation is just a whole bunch of that, right? Happens over and over and over and over again. And some proteins, particularly eukaryotic proteins, can be absolutely massive, right? 3,000, 4,000 amino acids in one long chain, right? So this can go on for a very, very long time. Some of them are super, super short. Like I think the shortest one that I know of is 7 amino acids, which is gonadotropin releasing hormone. It's involved in fertilization or something like that. Anyway, we've got to stop this point at some point, right? So if you do all transcripts, whether eukaryotic or prokaryotic, have a start codon and a stop codon. So the stop codon is either UAA, UAG, or UGA. You can see those down here in red. And now those don't encode for a tRNA called stop. They just don't encode for a tRNA at all. So essentially, once the ribosome comes across one of those tRNAs, those codons, sorry, there's no tRNA to go in there, right? And so here's our stop codon. Let's kind of zoom in a little bit. So this is obviously a very short gene. So this is the last codon, coding codon of the mRNA. So this codes for asparagine, right? So once this polypeptide is added to asparagine, the ribosome will translocate along one word, and now we will have a stop codon in the A site. So there is no tRNA which will now come in, right? So in and of itself, translation will stop, right? Because there's nothing to add this polypeptide to. There's no new amino acid in the P site. But also we need to release all of that various stuff. We need to release this polypeptide, and we need to release the ribosome from the mRNA, right? So the ribosome can be reused, kind of reassemble on another mRNA, and other ribosomes that are chunching along behind it can finish as well, right? And so that's carried out by the release factor. That will recognize an empty A site that's stalled over a start codon. And when it does two things. Oops. One, it cuts or catalyzes the release of the polypeptide, right? So that polypeptide is done, get folded, processed, whatever's going to happen to that. And then the ribosome is basically kind of pushed apart, right? And is ejected from the mRNA. Translation is over. Right? So quite often, translations kind of taught as like a one and done thing, right? Yeah, one mRNA, one ribosome goes along it, makes a protein. But really, it's actually a lot more complex than that. And that's because as soon as that start codon, and this is whether it's eukaryotic or prokaryotic, right? As soon as that start codon becomes available again, another ribosome can either assemble on it, if it's prokaryotic, or scan to it and begin translation. And so depending on the mRNA, and this varies from one gene to another, you can have potentially tens of ribosomes, even hundreds potentially, all chuntering along that mRNA producing polypeptides. This is called a polysome. And if you think about it, it's a pretty efficient way of making lots of protein, because you have to make X amount of mRNA, but for every mRNA you make by transcription, you can generate tens or hundreds of proteins. So it's a very good way of scaling up. And obviously this, whether or not this happens, is determined in large part by how efficient the initiation of translation is, at least in eukaryotes. It's controlled by other stuff in prokaryotes. And so it's about 25 amino acids, because these ribosomes are huge proteins, right? They're really big and easily visible. But as soon as that ribosome clears the start of the mRNA, the coding sequence, another ribosome can assemble and begin translation. Now, there's also some oddities with prokaryotes, right? So prokaryotes they're actually very interesting in many ways, right? And it helps to kind of think about the kind of context, right, in which bacteria exist, right? So bacteria can't control their environment. We can, got lights on, kind of nice temperature, somewhat fed, getting a bit hungry, right? Bacteria don't have all of those luxuries, right? So bacteria have to be able to respond to changes in their environment, particularly in terms of nutrition, right? Because they can move around, some of them can at least, but that also takes a bunch of energy too, right? So in terms of bacteria, what they tend to do is they group all of the genes that are involved in a particular process, metabolism of a particular nutrient, say like lactose is one that we're going to talk about. They group all of those genes together into one big kind of functional unit, right? This is called an operon. That's basically a collection of genes that are all involved in the same process and by and large regulated together. It's a little bit more complex in eukaryotes, but that very much applies in prokaryotes. And one operon equals one mRNA. And that's called a polycistronic mRNA. And what we mean by that is cistron is basically a unit of information, a coding sequence. So multiple coding sequences, ORFs, etc., on the same mRNA. Now, these each have their own start and stop codon, right? So they each generate a distinct protein. But they're all together in the same mRNA. And so that is super efficient if you think about it from a kind of organismal perspective, right? If you're on lactose and you don't have glucose, you need to express all of the genes involved in metabolizing lactose. There's no point just expressing one of them because you're not going to get any glucose, right? And that also means it's very easy to turn off all of those genes when they're not needed. And so remember again, prokaryotes, the genes are always on. They have to be turned off. And so this allows what's called coordinated gene expression. If I can spell that right. And that basically means all of the genes involved in one process are controlled together. right? And polycistronic mRNAs are essentially the result of that. Eukaryotes doesn't happen that way. We do have operons, kind of. They're a bit different, but eukaryotic mRNAs are almost always one mRNA, one coding sequence. And another kind of curiosity that I've mentioned already a time or two is that transcription and translation in prokaryotes are coupled. Right, so they occur in the same place. They're not separated by space as they are in eukaryotes, right? We have transcription in the nucleus, translation in the cytoplasm. In prokaryotes, it all happens in the same space. But also, it happens at the same time. So in prokaryotes, as soon as that mRNA emerges from the RNA polymerase, right? So these red blobs are the RNA polymerase. Start the gene up here, or operon towards the end down here. As soon as enough of that mRNA has emerged, a ribosome will assemble on it. I remember how long that is exactly. I think it's like 40 or 50 base pairs, give or take. And as we've already said, as soon as the ribosome has moved far enough along, another one will assemble on that start codon. So the mRNA is being translated as it is being made, which is kind of crazy to think about, right? So the mRNA hasn't even been finished, and we already have this polysome formed and proteins being produced from that mRNA. Right, and this allows a couple of different things. One, it allows prokaryotes to scale up gene expression very quickly. Right, so they're able to respond very quickly to changes in their environment, whatever those changes may be. And the second is that this actually ties in to a lot of how bacteria control gene expression. Right, so this allows some real funky tweaking of gene expression levels, which we'll talk about when we get to the lac operon and the trip operon, especially the trip operon, which are two different examples that we'll cover after the exam. All right, doing okay for time so far, so let's have a little pause, see if anyone needs time to catch up or has questions to ask. Not just questions about this stuff. If you have questions about anything else also, you know, because exams on Thursday, now's a good time to pipe up. Okay, cool. Well, I'll be around mostly the next couple of days. So if you do have questions that come to mind, or as you're going over the material, things that you'd like help in explaining better, give me a shout. Send me an email and I'll answer that as soon as I am available to do so. Okay, so last few things I want to talk about are the genetic code and degeneracy and wobble, which sounds a little bit funny. One of my dog's favorite or older dogs and most robust toys called a wobble ball, which is really cool. Made out of hard plastic. She still hasn't destroyed it, which is pretty impressive. Okay, so if we look at the genetic code, right? So this is, get you down there, what it looks like. I'm going to move these things over so I can see what I'm looking at. Essentially, you have 64 different codons. 3 of those are stop codons. 1 of them is a start codon. Right? There's only 1 codon for methionine, and you don't often see it in proteins. Do sometimes, but is always at the beginning and much more rarely elsewhere. So we have 60 other codons, right? And that's shared between 19 amino acids. Right, so we have a lot of redundancy, right? And this is a good thing. This is a good thing because essentially you can have mutations in a coding sequence that don't change the amino acid sequence of the protein. And so if you look throughout this table, and the way you read it is this is the first base, the one along the top is the second base, and then you'll see the third base kind of actually in there. Right, so you look here, proline, for example, has 4 different codons, right? And so whether or not in the mRNA it's CCU, CCC, CCA, or CCG, you'll get a proline added to the polypeptide in that position. And so what that means is that as long as you have a CC, you'll get proline, sorry. This is called degeneracy, right? It means that more than 1 codon codes for 1 amino acid. Right, and that gives us essentially, or organisms, a bit of a buffer against mutation. Now, if the first base is changed, totally different kettle of fish. So if the first base is changed to a U, for example, then we have a serine. And that is a change. That's actually a fairly decent change. Let's see, can I go all the way back up here? So proline from memory is a hydrophobic. Oh yeah, there we go. It's actually kind of a funky looking amino acid, right? So if this is changed from a proline to a serine, cysteine. That's what I was talking about. Oh, cysteine, here we go. The cysteine, that goes from hydrophobic to hydrophilic, right? Also somewhat large to somewhat small, and these can form disulfide bridges, right? So if that change occurred, it's gonna whoosh down here. Oh no, I was right, it was a serine. Even so, same applies. If that occurs, then that's a pretty fundamental change in that protein. And most likely it depends on where in the protein it is, that would change the function of that protein. Usually reduces it or gets rid of it entirely. So we can have single base changes within the genetic code and not have a change in the amino acid sequence. What we absolutely cannot have is additions or removal or deletions, insertions or deletions of bases into the open reading frame, right? Because the reading frame is entirely determined by the start codon. So wherever the start codon is, the reading frame is from that point. So the next word is UUU, the next is GAA, GUG, AGU, so on and so forth, right? So the ribosome will read the mRNA in groups of 3 bases. It doesn't know what those codons should be. All it knows is that there's a start codon here, and it will read, because of the structure of the ribosome, every 3 bases from that point. So if you introduce a base, or if you remove it, you change that reading frame. So if you removed that U, we're looking at just here. If you remove that U, then the reading frame would now be UUG, AAG, UGA, GU something. That's called a frame shift mutation, and that is bad. There's kind of like a cute little way of looking at it here. Right, so if we had, whoever did this had a very good grasp of 3-letter words. But if you have a sequence of words that make some kind of sense, right? If you add a letter to that sequence, you shift the reading frame 1 base. Actually, this is, I've got to look into this some more. This is how COVID virus is translated. Funnily enough, it has 2 reading frames, and they're separated by 1 kind of base. So somehow, I don't actually understand how it's able to shift between those 2 and generate different proteins, which is pretty wild as far as I'm concerned. So if you mess around with the reading frame, typically what you get at the very least is a different protein, right? You'll end up with a different sequence of polypeptides. And that's bad, right? So you're going to generate a protein which makes no sense. It's called nonsense, actually. It's a nonsense mutation. Also, what you will find is quite often, here's one actually. UGA. What is UGA? UGA is, too zoomed in. So stop codon. So frame shift mutations not only result in gibberish, they will also result in a truncated protein. Because if that frame is shifted so that now this UGA is in frame, this constitutes a word, then when the ribosome gets to that point, it will stop translation. It will be terminated by the release factor. Right. So frame shift mutations are very, very bad, and they will almost always result in a non-functional protein, a complete loss of function. So essentially, there's kind of 2 different types of mutation. Single base pair changes, depending on where they're at, may have absolutely no effect. Single base pair additions or deletions can have a huge effect. All right, so just to wrap up, just to kind of follow on from that talk about degeneracy and wobble, kind of, I haven't exactly told you a porcupine, but I've not entirely been super precise, right? Anybody watched Austin Powers, the films? Well, Austin Powers is supposedly a Cockney. And so my parents weren't Cockneys, but they grew up in East London. And Cockney rhyming slang is funny. So porcupine is lie. There you go. Learn a little bit of Cockney rhyme and slang today. My favorite one is trouble and strife. Can anybody guess what that rhymes with? Wife. Anyway, there's a whole bunch. Another favorite one is septic tank, is rhyming slang for yank or an American. So if you ever go to East London and you're called a septic, that's why. And there's also other things. So septic tanks tend to be full of. Anyway, I should probably stop insulting my class, especially just before an exam. Anyway, those are kind of some of my favorite ones. And because my parents grew up there, this is just kind of stuff that I kind of absorbed as a child, I guess. So there aren't as many tRNAs as there are codons, right? If you think about it, there are 61 different codons. That'd be an awful lot of tRNAs and amino acid tRNA synthetases. So typically the third base is not super important, right? So you can see here with proline, for example, the tRNAs for proline recognize C and C in the first and second base. The last base is much less important. That's why we have that degeneracy. As long as there's a C and a C, the tRNA will come in and you'll get proline added in that spot. And so the degeneracy and wobble really kind of go together essentially, right? So tRNAs are very specific, but they're very specific for not entirely all of the codon. Now methionine tRNA is, right? If there's an AU there, it has to have a G in the third spot. So methionine, the methionine tRNA is very specific. Isoleucine one, not so much, as long as it's not a G. Right, so there is variation in specificity between tRNAs as well. And that's pretty much it. So kind of hit it a few minutes ahead of time. Don't think I really have anything else to add. I could probably try and remember some more Cockney rhyming slang. Oh yeah, tables and chairs, stairs. It's really weird, actually. I mean, I haven't been to East London for a good long time, and I'm by no stretch a Cockney. In fact, I'm not even a Mockney, which stands for mock Cockney, like Jamie Oliver. Can't stand that guy. But anyway, yeah, tables and chairs. It's like an entire other language. So if you fell down the tables, it means that you fell down the stairs. Jimmy Riddle. Actually, my dad always uses this. I'm going for a Jimmy. Jimmy Riddle rhymes with piddle, and piddle is slang for going for a pee. So if you say you're going for a Jimmy, it means you're going to go to the toilet. There you go. A little bit of culture in your genetics class as well. All right, anybody have questions? Stuff to be clarified. Exams pretty much exactly the same as 1 and 2. Main differences are going to be lots of details. Well, Michael, if I knew what the class average was before I set the exam, that would be amazing. I'll be telepathetic, as my mum would say. This upcoming exam, yeah, I've got absolutely no idea what the class average for the exam 3 will be. Exam 2 and 1 have been okay. I think the class average has actually been a bit above 70, give or take, which is pretty good. Yeah, it will be harder in the sense that there will be more details, right? As you've seen throughout all of this, there's a lot of stuff going on. It'll be easier in a sense because there's less calculations and stuff that you need to do, right? So you won't need to work out any crosses. Do we do 3-point crosses? No, we've done that already. Yeah, that was a ways ago. And so those kinds of more time-intensive questions won't be there, but there will be equivalent match of kind of more complex questions that I'll give you the beginning of the information and I'll give the end, and I'll expect you to join the dots together to get to the right answer. And a whole bunch of basic recall as well. So, you know, if you're on top of your stuff, it shouldn't be terrible, but you really do have to make sure you know your material, because there's not going to be enough time to look it all up. That's pretty much it. Cool. All right. So if any of you have any questions, please do feel free to get in touch, and I'll either see you in about 18 minutes or I'll see you some other time. Oh, I also will be in kind of Zoom at this kind of time point, give or take, for the start of class, in case you have any questions or difficulties with the exam. So I'll be there for, I don't know, it depends how long I can stand sitting in the chair just reading the news, but I'll be here for a little while, at least, probably about 45 minutes, give or take. Cool. All right. Good luck, everybody. Work hard. I'll see you soon.